Welcome, everyone. My name is Ulrich Broeckel, and I'm the founder and CEO of RPRD Diagnostics. I want to welcome you all to our presentation focusing on genetics and polypharmacy and the use of pharmacogenetics to better the management of drugs and improve quality of life, particularly for the elderly. We obviously had planned to meet in person and that I could give you this presentation live, but given the current circumstances, I really want to thank the organizers again for giving us the opportunity and give this presentation as a recording. So again, I want to thank the organizers for inviting us. And in my talk, I would like to give you an overview of the work that our company does and what we're focusing on, particularly focusing on how pharmacogenetics and genetics can improve the medication management in patients in general, but particularly also for an older populations and senior citizens. If we're thinking about polypharmacy in the elderly, I want to give you a little bit of background, and I want to introduce you to the concepts of pharmacogenetics and show you some of the applications, some of the examples where we think pharmacogenetics can improve the treatment and how we manage medications in patients. So let me give you a background of how frequent it is that patients actually receive medication where testing could potentially be beneficial. So if we're looking at older adults, and that will be defined as individuals over 65, about 39% of older adults receive more than 5 prescribed drugs. In addition to that, about 72% of older adults also take at least one over-the-counter medication. And the other data shows from publications, numerous publications shows that about 59% of older male patients take one or more unnecessary prescribed drugs. With that background, when we're thinking about this, how many drugs are prescribed to patients, obviously we're prescribing these drugs to treat a disease and improve the outcome. But what also happens if you take a medication doesn't work for every patient uniformly for all patients. And it is well established that some patients actually experience what we call adverse drug events. And adverse drug events are defined as an injury resulting from medical intervention related to drugs. And if we're thinking about adverse drug reactions, they are the non-responder or the occurrence of side effects, medication error, allergic reactions, overdose, among all, a few other examples here. Now, when we're thinking about how often this can happen, what this graph shows here is that the risk of adverse drug events increases quite significantly the more medications a patient receives. So we're looking, for example, at a patient who receives 2 medications, the risk for adverse drug reactions and events is about 15%. And it really increases then if we're looking at patients, for example, who receive 5 medications. And it is quite significant if we're looking at patients with more than 7 medications. About 82% of patients receiving 7 or more drugs have a very high likelihood, about 82%, of experiencing adverse drug reactions. So this really represents, particularly in senior citizens and patients and elderly patients, that presents a quite significant clinical problem. Now, how do we think that pharmacogenetics can actually benefit and manage the patient and the medication better? I want to introduce you to a number of terms. So when we're thinking about how drugs work, we need to look at the response of the body, what a drug does to a patient, and that is really the field of pharmacodynamics. So that describes the biomedical and physiological effects of a drug on the body. So what does a drug really do to a patient or a patient's body? Pharmacokinetics then describes the movement of a drug in the body as it moves through the different organs to reach the particular organ where it has to exert its effects. So what does the body actually do with a drug? That also includes the effects of like how a drug gets metabolized and excreted. In that area, then, pharmacogenomics really looks at the impact of genetics, how genetic variation, the variation in our DNA, what we inherit, how that affects the response to a drug. So what does a gene do to the drug in a particular patient? So this is really the area what RPRD diagnostic focuses on. We try to bring pharmacogenetics, and we use pharmacogenetics to improve the drug management. When we think about what parts of the body really contributes to the effect and what target organs play a role and where genes play a role, to better understand then what genes we're actually testing, it's probably, it's a good start to think about where, what's the path of a drug when a patient actually takes a drug. So the first thing is, if you just take a pill, here on the left side, you see the first step is obviously the absorption. It goes in our stomach, it goes in our intestines, it dissolves and actually goes then in the bloodstream. Then from the bloodstream, the first pass is for most of these drugs, they go to the liver. The liver, in many cases, does a first step of metabolizing, activating the drug. It also, the liver plays a big role in detoxifying and preparing a drug for the excretion then. But once the drug gets to the first pass, it gets circulated in the whole body, in the blood vessels, and it at the end reaches the organ which the drug has to exert its effect. At the same time, as it circulates, drugs also get then excreted. They either get metabolized again to the drug, so they become inactive, or they get excreted in the kidneys. And where many drugs get excreted then in the kidneys is the elimination and the transport from the bloodstream into the urine. So this is really the path most of the drugs take, which we ingest as a pill or even as an injection. And pharmacogenetics now tries to understand how genes really related to these mechanisms which I just described. So the absorption, the transport, the uptake, the metabolism of a drug in the liver, and also the excretion in the kidney, for example, how these genes and the variation in the DNA actually overall contributes to the effect of a particular drug. When we're thinking about pharmacogenetics, and this is really now more from an epidemiological perspective, we all think, that a drug should work in every patient about in the same way, but that is obviously not the case. Hence the examples of adverse drug reactions. If you here look on the left side, if we're taking all patients with the same diagnosis, for example, and they receive all the same drug, then a subgroup of these patients here are, for example, non-responders. So these patients do not respond to a drug, or they actually see an effect, the drug actually works, but for a number of reasons, including genetics, patients actually see toxic effects. So they have a much higher likelihood of seeing adverse drug events and toxicity. And pharmacogenetics really then, the goal of pharmacogenetics is to separate and remove and recognize patients who are either non-responders or who have a high likelihood to be toxic responders, who have a high likelihood of not benefiting of a drug, and really only then identify and treat patients for whom this drug actually has the strongest effect and the most benefit. So what factors actually contribute to this? Obviously, as I told you, it's the uptake, it's the absorption, it's the distribution. And these genes get categorized in the group of ADME, ADME genes. We're also looking at genes where a drug, for example, has to bind to a certain receptor in a particular organ, or actually also inside a cell where it exerts certain functions. For example, it targets certain parts inside a cell in an organ. Other factors, and I need to mention this, it's not only genetics, obviously, other factors which contribute to the overall effectiveness or the occurrence of adverse drug events and toxic effects is obviously the age, is diet, is the chronic underlying disease, kidney disease, liver disease, and so on. These are all really well-established factors. And if you think about this, physicians actually, we've been used to including the knowledge about these factors in prescribing drugs. So certain patients, for example, with a insufficiency or a reduced kidney function, we're actually well aware that we have to, for example, decrease the dose. And so by analogy, if you have certain genes which contribute to the effect of, as the kidney plays a role in excretion, if certain genes also affect that, it would be, and it is very logical, then adjust the dose of a drug similarly to what we see with reduced kidney function. Now, how frequent actually is it that a patient or patients can benefit from pharmacogenetic testing? And we have actually pretty strong evidence and data from a lot of genetic studies which look at the frequency of genes or gene variants, pharmacogenes, which are actionable, where if you have the certain variant, you would actually have to adjust the dose. And if we're looking at the overall prescription in the US, which are patients prescribed every year, about 18% of all the prescriptions filled every year in the US have links to pharmacogenes. And the other really important point to make is that if we're looking at a population, healthy population, patients with disease, and if we're looking at the most frequently tested pharmacogenes, then about 90 to 99%, which basically means pretty much everyone, if you're listening here, has at least one gene, one pharmacogene, where if you receive a drug which is linked to your pharmacogene, that actually a dose or the drug should be adjusted, or you might have to pick a different drug. So it is pretty frequent. Pretty much everyone has a pharmacogene which is actionable if you receive a certain drug. So pharmacogenetics is really very relevant, not only for a subset of patients, for a small group of patients, but obviously for the population at large. Now, when we're thinking about who would benefit from pharmacogenetic testing and what are the reasons when we would recommend pharmacogenetic testing, there are a number of different approaches which have now been well established and which we are pursuing as our company who's in the space of pharmacogenetics and clinical testing. So first of all, I think it might be indicated for patients taking medication who have experienced adverse drug reactions. If you're interested in figuring out or excluding or investigating this further for certain drugs, then pharmacogenetic testing would be recommended. We also think that patients taking multiple medications, and the word for that is polypharmacy, who see unexpected effects, for example, treatment failure or unusual unexpected toxicities, pharmacogenetic testing could help to further understand these effects and potentially adjust the treatment, either by adjusting the dose of a medication or switching to different doses in different drug classes. The other reason why pharmacogenetic testing can be recommended is for patients with a positive family history of adverse drug reactions. So there are other family members, and there are some rare diseases where if you have family members who already have an adverse drug reaction, there's obviously an increased likelihood that a patient also, because you inherit your genes from your parents and genes travel in families, that a pharmacogenetic test can be recommended because of the increased likelihood of also having adverse drug reactions. And then lastly, patients who need high-risk medications or drugs where there is what we call a very narrow therapeutic window or the potential for serious side effects. A narrow therapeutic window means that a certain drug a certain drug needs a certain drug level, and the target drug level and at the level where you see side effects is very, very closely linked. Normally, drugs have more or less a wider therapeutic window, but some drugs have a very narrow window, and I'll show you some example here in a bit, and where drug pharmacogenetic testing can actually be beneficial. All in all, there are really two modalities how you can do pharmacogenetic testing. The one thing would be considered reactive. Those are the first three examples where you have a positive family history, you've experienced unexpected effects, or you have adverse drug reactions. In response to what we observe, we would recommend pharmacogenetic testing. Now, for patients with polypharmacy, and as I mentioned this at the very beginning of my presentation, that's very likely in patients and elderly patients, in senior patients who have many or receive many different medications. Preemptive pharmacogenetic testing could actually be recommended. So you do pharmacogenetic testing. All the results are available for a physician, and as you adjust and physicians adjust the medication regimen, they can use the pharmacogenetic information before they prescribe a drug and before a patient potentially sees an adverse drug reaction and, for example, has a treatment failure. The advantage of preemptive testing is also that you really don't have to wait for a pharmacogenetic test. In many cases, sometimes tests take up to a week or 2 weeks. Many times patients need to receive a medication right away. And if you have a very comprehensive all pharmacogenes available testing in the medical record, then this information is available to the treating physicians right at the time when a physician has to make a decision what type of drug he or she needs to prescribe. So the benefit from preemptive pharmacogenetic testing, and this is the concept we as RPRD Diagnostics pursue and advocate for, is that it reduces adverse drug reactions, resulting in fewer emergency room visits and a lower mortality, and that's been supported by a number of different studies now. For certain drugs, it reduces the length of stay, it reduces lower readmission rates, it reduces the total cost per discharge, and it also improves the patient's drug adherence. Drug adherence, I think, is a very significant part in medication management. Patients who often experience adverse drug events are very hesitant to take their medication, and trial and error, figure out which is the best medication for a patient can be very frustrating, not only for the patient, but also for the physician. So improving the overall management and the selection of drugs will at the end improve the adherence of patients and increase the overall satisfaction. So I want to give you now some examples for what drugs pharmacogenetic testing is recommended and where we have clear guidelines. There is a consortium called CPIC. This is the Clinical Pharmacogenetics Implementation Consortium, group of physicians and scientists in the field of pharmacogenetics who establish guidelines and review the evidence for clinical testing. And so the examples what I'll show you are, there are guidelines available for physicians who would like to dive deeper into this and want to learn more about this. But there's a broad variety of drugs where pharmacogenetic testing and pharmacogenes are linked to their response or the occurrence of adverse drug effects. That's the group of anticoagulants, for example, blood thinners. There are many drugs in the area of antipsychotics and antidepressants, where pharmacogenetics can substantially help to select the best drug. Statins, those are the drugs to lower cholesterol, but also drugs against antivirus or antifungal medication against fungal infections. So it's a broad spectrum. It also covers all specialties. It's not only internal medicine, it's not only geriatrics, it also covers gastroenterology, cardiology. So it's truly a multidisciplinary focus. So I want to give you some examples where pharmacogenetic testing has been shown to be beneficial. And I want to show you some examples for drugs which are quite commonly used. And the first drug is a drug as a blood thinner. It's warfarin. It is still the most widely prescribed oral anticoagulant in the US and also Europe, worldwide. Patients who are receiving this are patients after a thrombotic event, but also patients with other underlying conditions where anticoagulation is necessary. And on the right side, you actually see it's decreasing a little bit, but there's still a significant number of patients in the US who receive this drug. Now, warfarin was one of these examples where very early on we recognized that a number of genes contribute, 2 or 3 genes actually, contribute to the overall response to this drug. And warfarin is also a drug with a very narrow therapeutic window. We try to reach a certain level of anticoagulation in patients, but obviously if you increase the dose, and this is just sometimes in patients a very small amount, you have too much anticoagulation and you increase the risk, or you have the risk of internal bleeding, which obviously needs to be avoided. Here on the left side, you can actually see this is a plot from a genetic studies where investigators scanned and screened all chromosomes, the whole genome. And so on the x-axis, you see all the different chromosomes. On the y-axis, you see different markers in the genome. And we see highly significant association or evidence that 2 genes, CYP2C9 and VCORC1, in addition to another gene called CYP4F2, substantially contribute to the variation in the particular dose a patient needs to receive a target anticoagulation. Warfarin response is a highly heritable trait, so it's highly determined by genes, and it is also highly variable between different individuals. And genetics can explain a large part, and pharmacogenetic testing can explain a large part of this variability. And if we have this information ahead of time, then you can start dosing a patient much more targeted, and you avoid the occurrence of potential life-threatening side effects. Here's an example then for a follow-up, a publication published a few years ago. When you look at patients who receive pharmacogenetic testing before they get treated with warfarin and the onset, and their dose gets adjusted based on the genetic testing, and if that can be compared to basically a group of patients who just receive the drug and get monitored, and pharmacogenetic information is not included. You can see here on the left side here in the intervention group, this is kind of the reddish brownish. You see a significantly reduced reduction in hospitalization compared to the historical control group. And you can also look at this more specific if you look at the rate for hospitalization due to bleeding, which is a well-established complication. If the warfarin is dosed too high, then patients who have the pharmacogenetic information which is used to adjust the dose, they also have a reduced risk of hospitalization due to bleeding compared to the historical control. So this is a very important study published in 2011, which really shows the beneficial not only in targeting and bringing a patient faster to the treatment target, but also in clear outcomes, which obviously affect the overall cost of healthcare. Now, how do we test for this? And this is really a very complicated slide, a workflow for somebody who want to dive deep into this. If you want to understand this, but it's been established now. It's really not that. It's really well established. There are clear algorithms physicians can follow when they have pharmacogenetic results. So depending on the result of your genetic testing, there are clear steps you can take. There are clear recommendations for physicians to adjust the dose to overall improve the treatment target and how quickly you can reach the treatment target. I want to show you and talk about another example. Also a widely used drug here, is widely prescribed over many years now. The drug is called Clopidogrel, or the trade name is Plavix. It is a very commonly used drug. It's a platelet inhibits the platelet aggregation. and is often used in patients after a stent when you had a heart attack or a narrowing in your vessels which support your heart muscle, and that stent is put in, then patients receive this drug. Clopidogrel is a very interesting drug. The drug itself, when you take this as a pill, is actually not active. It's called a prodrug, and it actually needs to be activated in your liver. So your patient takes the drug, it gets absorbed, it goes into the liver, and the liver actually activates this drug, and then it actually makes it into the bloodstream. We understand the metabolism of clopidogrel Plavix quite well. In the liver, there are a number of genes in the liver involved in activating the drug before it gets excreted, before it goes to the blood platelet, and where it exerts its function. Now, a few years ago, similar to warfarin, we identified a particular gene, and a variation in this gene, which affects the activity of the enzyme which activates the drug. And as you probably can guess, an enzyme which is, for example, non-existent or has a very reduced activity, then there is not as much. If all patients receive the same dose of clopidogrel, they do not have, they have a reduced amount. If you have a reduced function, you have a reduced activity, and you have reduced active drug in your bloodstream. And is that actually really relevant? And this is the study here, and this was a study published now over 10 years ago, which shows if you're a carrier or if you're a non-carrier, you can look at the efficacy, but you can also look at if you, for example, received a stent in a coronary artery, that carriers of a reduced function have a significantly higher risk of having another occlusion or another heart attack after a stent. And so patients who have a reduced metabolism, have reduced activity of the enzyme, have a reduced drug level, and have increased risk of cardiovascular events. Now, there are a number of other drugs you can replace that for patients who have this certain genetic makeup. And now it is probably widely recommended to actually do some testing before you prescribe this drug. And here again are the guidelines from CPIC, from the Clinical Pharmacogenetic Implementation Consortium for clopidogrel. Depending on your activity of your enzyme, whether you are like an ultra rapid metabolizer or if you are a poor metabolizer, we do recommend the adjustment of the dose or the actually switching to a different drug. So again, there are very clear examples and recommendations. If you are an intermediate or a poor metabolizer, your liver enzyme does not work or doesn't work as well, then an alternative drug, alternative drugs are actually recommended, because the drug itself, clopidogrel, does not function and you don't get the required level of anticoagulation, which would be necessary to prevent the occurrence of side effects. So that is another example in the anticoagulation field where pharmacogenetics can clearly contribute to improving the outcome and reducing adverse drug events. Let me give you another example here, we're moving away from the cardiovascular and anticoagulation field. A number of drugs are also linked to damaging the liver itself. Hepatotoxicity is one of the most common reasons for termination of drugs in clinical trials, and approximately about 33% of these terminations are due to toxicity of the liver. They're also contributing to the withdrawal of drugs from the market, and quite a number of genes are actually, or a number of drugs are actually linked with certain genes. So I'm picking here just another example here, which is linked to a genes called HLA, and where we're recommending adjusting the dose, or where this really directly affects the liver function. So that is another example where I think pharmacogenetic testing actually can play a role. So in conclusion, I would like to say that adverse drug reactions associated with polypharmacy are a significant problem. We see this particularly in elderly patients who receive, who have a high frequency of receiving multiple drugs. drug interactions are a significant problem, and obviously genetics also contributes to this. Genetics can, and pharmacogenetics explains in part the variation to the drug response among different individuals. This is the field of pharmacogenetics. And we do think that elderly patients and patients who receive polypharmacy can benefit from, particularly from preemptive pharmacogenetic testing, where you can adjust the dose and pick certain medications and look at the impact of genetics before you really prescribe a drug. There are more and more guidelines now becoming available, which are published, are available to the scientific and to the healthcare community, which annotate and present the evidence for this. And so I think there's a growing recognition that pharmacogenetics is quite important in selecting drugs and adjusting the dose. And using pharmacogenetic testing and adopting pharmacogenetics clinic guidelines really have the potential and to reduce adverse drug reactions and improve the quality of life. And I showed you some examples where we can clearly show that it reduces adverse events and can improve treatment. And so with that, I hope I gave you an introduction in pharmacogenetics. I hope I could convince you that pharmacogenetics can play an important part in the medication management for all patients, but particularly for older patients with polypharmacy. And I thank you again for your attention. And then I will be happy in a other segment to actually answer some questions. Thank you very much for your attention. And then we'll go forward and answer additional questions.